I understood sort of a school. You could almost call it a summer school today with the weather. And of course, a school then means also going into details. And so I'm going to actually concentrate on introducing a method that's very powerful for looking at molecular interactions and motion. It's a method that there has been no workshop about. I haven't even heard it mentioned in one talk so far. But I do agree that one needs to use multiple methods. And so, of course, I do multiple things. But I'm going to talk about one particular method. We'll get to it in a moment. And I'm going to go through explanations of how it works and some of the advances we've done with that. And I leave the amazing, awe-inspiring science to my colleagues. So we start out discussing, let's say, start out at the beginning, thermodynamic fluctuations. So, good, we have a time delay between Eastern and Western Europe. Okay. So here we go. If you look at particles on the nanoscale, they fluctuate. And these are 40 nanometer beads just undergoing Brownian motion. Now, if you want to analyze something like this, well, you can count, if you can count fast. And in fact, this experiment was done over 100 years ago. There had been stipulated by Einstein, these thermodynamic fluctuations, and so they actually set out to measure them. And they measured here the number of particles, gold nanobeads, observed as a function of time. Of course, well, what do you do with this information? It's actually very noisy. And so sometimes you have something important you want to communicate. Your molecules are trying to tell you something. And there's a message that's coming on, right? So you have this information, and what your dog hears is blah, 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 ginger, blah, 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 ginger. And what you'd like to do is you'd like to get the information out that you would like to have. It's, okay, Ginger, I've had it. Stay out of the garbage. Do you hear me, Ginger? Stay out. And so we do this with mathematics and with the correlation function. So we can plot here the numbers I showed you before. We do this mathematics, and we're not going to go into it in too much detail. I tried to just tell you the things that are important for you to know. But the thing is, you should realize math is your friend. I realize most of you have traumatic experience from your math teachers in high school, but you can do it. Math actually helps you. Okay, so we take our signal and we measure it then as a function. We take a copy of our signal here as a function fluctuations, we multiply it by itself, and then we just do a little bit of mathematics, or we're looking at the fluctuations on the signal. And there's two important things that we assume. One is that the signal is stationary. So your average parameters are not changing with you in time. Otherwise, you're kind of messed up, right? If the beginning of the experiment has different parameters than the end of the experiment, it's difficult, so we assume that it's, at least on the time scale of the measurements, the parameters are constant. And the processes are ergodic. So every sizable sampling of your process is representative of the whole. So it doesn't really matter if we measure one position for a long time, or we measure a lot of positions for a short period of time, or for one instance, we should get the same information. So in the case of fluorescence correlation spectroscopy, here you have some sort of noisy signal. Now we turn the noise into the signal, so we have something to measure. We have our curve, we make a copy of it, we start shifting it. Now, if you heard the nice talk from Thorsten Wohland, he had better animations. But you end up here looking at the self-similarity of this curve. And from that, you get out some function, and the function has very important information over the size of the fluctuation, so how many particles are in the fluctuations and how long these fluctuations persist. If you look at this function, these little brackets, if you think, what in the world is that, some sort of mathematical thing? Well, it means the average, which means you sum over all of the points you measure. Typically, we are in the digital age, so everything is now been digitalized, so you have a particular number of photons at every time point. You subtract that from the average value, so you get the fluctuation. You multiply that by the fluctuation with this delay. You sum it up, and you take the average. And that's important, we'll come back to that. It's also, if you take what the definition is, if you take no delay, what you actually get is a familiar equation. The top is the variance, the bottom is the average value squared. And if you have a Poissonian process, the variance is the average number. And so that's why the maximum, the correlation goes as 1 over the number of particles you have. And as you shift it out, you lose the correlation, and it goes to just purely random. And that's also nice, because of course things will purely randomly correlate, and you don't want to have to worry about that, and the mathematics takes it out for you. It says, okay, I know for this concentration, you'll have this number of random colocalizations. Now, we've seen a lot of beautiful images, and I agree that some of them have been alive, but usually when we show you these beautiful images with multicolored labeling, DAPI staining, microtubules, mitochondria, the cells are dead. They've been labeled to death. Okay. I'm interested in dynamics. Okay, we saw gold particles can be dynamic, but we'd like to look into cells. So life is a dynamic process. We'd like to look at it. Okay, so let's look at something that's dynamic and moving. So let's label. Well, we're going to zoom in here into a small area. And now we'll do a scan in this area. And a single GFP freely diffusing in this cytosol looks something like this. Not quite as awe-inspiring as the image before. Okay, we can make it green. Okay, that's better, right? Good, but it's dynamic. There is a lot of information in this image because the cell is still alive. And so how do we get the information out? Well, we could do a measurement at a single location. So you just measure the intensity in this spot for a function of time, and you can do a normal fluorescence correlation spectroscopy measurement. But if we have an ergodic system, the same information should be distributed over the spatial domain. So we could actually look at the spatial distribution of these fluctuations. And so to do that, this was introduced by Paul Weissman. He was in Niels Peterson's group. They introduced this image correlation spectroscopy. So you can imagine you have an image. You have here, a number of particles, people at the party last night, and you have an image. You make a copy of that image, and now, as in FCS, we had one dimension, time. We took a copy of it, and then we shifted with time to see how similar they were. Well, with an image, we have 2 dimensions, so we have to shift with 2 dimensions. So we have to shift in X, and we have to shift in Y, and so we call these shift parameters the C and phi that we shift. So we shift the picture a little bit. We see how self-similar it is. Then we shift it a little bit more and see how similar it is, and shift it a little bit more. And what happens? So if you have here something that's spread over a few pixels, like your point spread function, and you move a little bit, well, you decrease a little bit in intensity, you move a little bit more, it decreases some more, and you'll end up with something that looks like your particles. If the particles are bigger than your point spread function, it will give you the shape of your particle. If it's smaller than your point spread function, it gives you the point spread function technically squared, because we have the intensity squared in our correlation function. But we can get the average number of molecules per pixel out of the amplitude of this function. Okay, I want to talk about dynamics. We have here a single image. You don't have dynamic information in an image, right? I mean, you might have seen me last night at the party. I was next to, some people, maybe Manu Magre. Now, do we interact or do we not interact? Was that a random colocalization, or does it happen that we interact at other times during the conference, suggesting that I might know him? Well, then it'd be nice to have more dynamic information. Are they actually moving together? And it's actually, for most of the images we take, there is dynamic information in one image because of the way we take it. So typically we take raster scanning images. So a raster scan means you take a line scan, you come back, you take a second line scan, you come back, you take a third line scan, etc. And so you start building your image up as a function of time. And so you are encoding in the position of that image information recorded at different positions in time. And so if we look, we have a pixel time, the time it takes, you measure at 1 pixel, you time at a second pixel, time at a third, you have the time it takes to go back and take the second line. So you actually have 2 scales of time information, the fast time axis and the slow time axis, which actually gives you a broader distribution of time scales to look at. And then there's a frame time scale where you go back and look at what happens. So how do you interpret this? So this is simulated data. Okay, if I have particles moving around, what happens? Okay, I'll hit them in one line. I come back, maybe I'll hit it, maybe I'll miss it in the next line. And so I get these sort of little smears. I then do this image correlation. So I make a copy of the image, I start shifting it, and I'll get to the average smear of my image. And then, well, you can actually put in the information, put in your model of what you have. For example, a diffusing molecule. We put in the parameters we know, how big the, what's the pixel size, what's the line time, what's the pixel time, what's our point spread function, and we can get, with the help of mathematics, the mobility of the molecule out. So what does this mean? If we have molecules that are moving slowly, then what happens when you start to scan, in essence, the molecule doesn't move very much. And you just get a copy of your point spread function. And again, because it's the correlation, you get the point spread function squared. So you just basically see how good your focus is. And so if you do a competition in your lab later, going back, who can make the the smallest focal volume, whoever gets the smallest PSF wins. If you have a fast molecule, it will leave faster, and so it decays faster. But there's a chance you'll find it further away. You'll never run into it if it's moving slow. If it's moving fast, you can run into this particle. And so this shape in 2 dimensions has information, 2 scales, slow and fast. And so I'm talking about Raster Image Correlation Spectroscopy, so that's the method that I haven't heard yet, RICS. And the one other point about this, the dynamic information, it's in one image. Now, the image doesn't have a lot of data points. I mean, okay, with these, big cameras, if we were to do the, use the 250 million pixel cameras, then maybe we'd have some. But if we take like 512 by 512, that's not a lot of data points. Not so much as an FCS measurement. So what we do is we measure maybe 100 frames, 300 frames, and average the information together. So we calculate a spatial correlation function for each frame, and then we average them together, which also has some advantages we'll get back to. Good, so what does this look like? If it's immobile, these are simulated data, then you'll get your point spread function squared back. If they start moving, it will start to collapse along the 1 dimension, and if it's moving even faster, then you start getting streaks with a little bit of a foot passing out in the other dimension. Okay, well, that's simulated data. What about real data? Well, here's 20 nanometer beads in a laser scanning confocal microscope. Here's the picture of GFP in the cytosol I showed you before. And here's GFP labeled to a protein of HIV, the GAG structural protein. And if we look at the correlation functions, well, the one for the 20 nanometer beads, that's kind of what we'd expect. I mean, if I stop the movie, you see, okay, I hit a bead, I get this streak, I come back, well, it's gone. Oh, I see another one here. And I'm basically looking at the persistence of these structures in my image, so I get this little streak coming out with a little bit of a tail coming away. Now, if I stop the picture from GFP, I don't see the streaks. Now, if I was an electron microscopist, I would probably, but I think most of us don't see it, but math is your friend, and the mathematics will pull it out for you. In the third case, we actually have 2 components. We have a fast-moving component, and we have a slow-moving component. The slow-moving component gives the round shape, and the fast one gives this collapsed shape. So then what often comes up is the question, when do I want to use FCS? When do I want to use RICS? Well, here we've done some measurements. These are in vitro measurements. We did it in buffer, and we did it in a viscous buffer that mimics the cytosol of the cell. The problem is that if you have too much laser intensity, you get photobleaching, and your trajectory stops short. But if you turn on the, increase laser light, you get more power. So if you look here, we're doing FCS. You increase laser power, the molecule gets brighter, that's kind of nice. Well, but at some point, even here, it starts getting worse. And if we look at the diffusion coefficient at 10 microwatts, the diffusion coefficient continually gets faster and faster. At 2 microwatts, it doesn't change all that much. If you do it now with RICS, then they're much more stable, because you're actually distributing the information over space. You're not always, drilling or burning a hole at 1 pixel in your cell, but you're distributing the laser power throughout the cell. You also have the advantage that you have an image. You can see what's happening. And if there's weird things going on, if there's the picture I showed previously with the GFP here in the cell cytosol, well, there's also vesicles in there that may change the correlation. So if I want a really good correlation, I might want to get rid of them. I can do that if I have the image. So if you don't need the fast time scale of FCS, or you don't want to try to fit 5 different species, well, the RICS is better off in cells. Usually. It's not always the case, but it's usually. Okay, what are the downsides of RICS? Well, the first thing is that RICS requires a square image. Why is that? Well, because to do it efficiently, you end up taking the fast Fourier transform, and that works well with a square box. Cells aren't typically round. Well, typically square. We can make them square. Some people have done that well, but the nucleus is still a pain and in the problem, so okay, that's not going to work. It's also very sensitive to outliers. Now, like I said, the intensity, the correlation goes as the intensity squared. And so if you have 2 species, things get weighted non-linearly, and that makes your life a little bit nasty. And what that means is if you get one bright object going through your image series, it totally messes up the correlation. It makes it really a pain. And so measuring samples that tend to aggregate can be very difficult. And then we get spatially average information. We're scanning over the image, and we get one diffusional coefficient for that image. Of course, again, if you heard the nice talk from Thorsten Wohland, he showed you now how with image ICS, you can calculate the diffusion coefficient and get these beautiful maps over the cell. So can we avoid these issues? So let's go back a little bit into the details of how we calculate this correlation function. And so we have, the fluctuation of the intensity in each pixel, and then we shift it. But if I have my image, I have the region I want to look at. Okay, let's say that I take the whole image for right now. But every time I have a 20 by 20 image, and now I start shifting. So I shift one image with respect to the other. Well, 1 pixel falls off, so I only have 19 pixels I can compare. And I shift it again, then I have 18 pixels I compare. I shift it again, I only have 17. So I have to renormalize to the number of pixels I have. So this funny bracket thing is a sum divided by the total. And this is the total number of elements we have. So, and if you actually look, this is what this region of interest looks like. If we just do the correlation, we get less and less. Now, I had a postdoc, Jelle Hendricks. He had then also this great idea. Why don't we just take a image, which we call our region of interest. We make 1s and 0s. We put 1s where we want to look at the object and 0s otherwise. And so we end up multiplying our intensity of the original image by our region of interest matrix, so to say, which is basically 1s and 0s. So we either get the signal if we want it, or we make it 0. And then we renormalize that to the total number of times I measure a shift of x by 1 or x by 2 or y by 1 or x by 1 and y by 1. And I normalize then correctly to the number of pixels in that image that gives me the correlation point in my image I would like to see. So how do you want to select the region of interest? Well, 1, drawing. You can just select by hand. Okay, this is the region I want to interest, what I want to do. You put in 1 inside, 0 outside, or 1 outside, 0 inside, whatever you'd like to do. You can do thresholding on pixels, however you'd like to. You can do an intensity threshold, so if something gets too bright, you throw it out. Or variation threshold, if the variation in the signal gets too bad, you throw it out. So you can use some sort of thresholding, or you can divide things. And so now all of a sudden automatically, once we get above 150 kilohertz, it recognizes bright objects, and those pixels then get thrown out of the analysis. We can use another channel, so we image the fluctuations of a protein in one channel. We may then put like a marker, mitochondrial marker, in a second channel, and then we have our region of interest that we want to look at. We multiply that then by the image, and then we can do RICS, arbitrary region RICS, to get out the diffusion coefficient on an image. Now, I mentioned that we calculate the spatial correlation on every frame of the movie. And so I can also do this region of interest on every frame of the movie. So I can have a dynamic structure. I have a structure that's moving, and I segregate every single image, and then just multiply that each frame by the corresponding image to get my region of interest. And maybe you want to combine some of these. So then you just end up taking the product of all the pixels that are 0s and 1s for the regions of interest you've selected, and then you end up with your final region of interest that you can do your arbitrary region with RICS on. This is all in our software, which is you can freely download. It's written in MATLAB, but you can also get the runtime or the source code. So does it work? Well, here's a solution of Venus, and we get these aggregates. So if you do a histogram of photons per pixels, you see the aggregates going through. And if you try to fit this, all of a sudden you don't have a single species, you have 2 species, you have the fast-moving species, and then you have a 37% of something that looks, we would call it immobile, meaning it's immobile on the time scale of scanning over that object, which is maybe on the order of 50 milliseconds. So in 50 milliseconds, if it doesn't change much, you get your point spread function. Good. If we now clean up the data, the aggregates disappear. We only take pixels where the average of the 5x5 area is less than 150 kilohertz, and the software recognizes these aggregates going through. If we look at the G0 values, then they behave instead of getting these spikes where you have the aggregates, and we can nicely see 100% freely diffusing protein with then a non-perturbed diffusion coefficient. Then you can do things like bacteria cells, where you look into the, you can select from the intensity what are within the bacteria, and then you can do FCS to look at diffusion of proteins with bacteria. Or now you can actually do nice, if you have an image of multiple bacteria, or even, I saw the nice image from Cecilia Fraden about in the Drosophila nuclei, you have all these space nuclei, then you can segregate your image and do RICS in the individual images. Then we can do some segregation. So here is the question of diffusion in the membrane versus diffusion in the cytosol. So we have a cytosolic protein, Venus. We have the membrane protein, which is a palmulated or methylated palm version of the of YFP. They're both spectrally the same, so they both show up in the same channel. The cytosol we can also look at with M-Cherry. Then we did here, Yella did a intensity threshold saying, okay, the membrane is above 300 kilohertz, the cytosol below, and then you can do your correlation within these regions of interest, and you get out the slow membrane diffusion for the membrane, 0.3 micrometers squared per second, and the normal free diffusion of a protein in the cell from the cytosol. And once you can classify an image, then of course you can take all of the pixels that belong to a particular classification, do this region of interest analysis, and then you can sort of plot out the diffusion coefficient on the various classifications. Okay, so the arbitrary regions RICS, it's generalized any region of shape. You can use non-perfect samples. I should say that it's, of course, always better There's no substitute for good data. And no matter all the cute methods we develop, you'll always want to take the best data possible because that will always push the limits of what you can observe. But in some cases, you don't have to throw the data away. You can still do a very good job. We can parallelize experiments. We can look at RICS in organelles, outside of organelles, and do pseudo maps. And there's a nice paper here by Jelle Hendricks. It's not my paper, but they did one that'll tell you what are the optimal parameters depending on what you want to look at for RICS. Okay, so just to show you that we can apply this to science, we did a collaboration with Mary Elena Torres-Valdile from the Helmholtz in Munich, just across the street. So two of my students worked on the project. So they were looking into condensates development during mouse embryo development. So we got mouse embryos with the 2-cell stage. And these here, it's stained with DAPI. You can see these nuclear-like bodies. You have this pericentrosome region around them, and at the early stage they form these sort of nice little circles, and then they, at the 4-cell stage, move into these chromocenters. And the question was, do we actually have condensates forming in these chromocenters? It's been seen in Drosophila. Do we see this also in the mouse embryos? Now, condensates have been a big thing in the last several years, and a lot of people have looked at them in vitro, and you get these beautiful large differences between the properties of a condensate inside and outside in vitro. But if you go into cells, the inside and the outside of the condensate aren't all that different. And so we applied a number of different criteria to try to figure out, are we in the condensate, are we not, or is it a condensate, is it not a condensate, is what we wanted to answer. And so we used looking for fusion and fission events, change in sphericity over time, changes in volume. What about the diffusion inside, diffusion outside, and are there boundaries? So if we did just imaging, I mean, we can take the same images that we were using for RICS and just look at them, and you can then see events where you have fusion or see events where you have fission. So yes, these things are changing their shape and structure with time. We could then measure their how spherical they are. We could measure their volume. They're very similar at the early 2-cell stage and then diverged as they went towards the 4-cell stage. And then we could use this major satellite repeat marker, which goes to the pericentrosomes, to determine what regions are in the centrosomes, what is inside and what is outside. And then we looked at the diffusion of the histone 3.1, use the arbitrary region RICS to analyze this. And so you can see in the nucleus outside, you have a faster diffusion than you do inside. If we now look at the 4-cell stage rather than the 2-cell stage, they get much closer together, and the number slightly changes between the 2-cell and the 4-cell stage. So there is a difference in diffusion and also a difference in concentration, suggesting there is a phase separation between them. And then we also did FRAPT to look at it. Again, there's not huge differences, but there is slower kinetics we have slower kinetics exchanging, it's suggesting that we do have the presence of a boundary filling up. So indeed, we can look into differences when we, using this arbitrary region RICS among other things. Now, we talked about using the other channel, but now what we could also do is we could do a dual color cross correlation, dual color RICS, right? So you can imagine now we have 2 labels. So you can see, I'd say, we can separate men and women. So we have the men, we have the women. I guess, okay, men were blue, women are yellow. We do the correlation. And so we can look at autocorrelations that look at the men. We can look at the autocorrelations that look at the women, right? So I mean, I give the analogy of taking the security cameras in the grocery store, and you can just now with facial recognition determine those that are men and those that are women going shopping. And you can see how long men take to shop. You can see how long women take to shop. And I better not make a statement about who I think is faster, not to avoid any potential sexist comments. You can ask me later after a beer. But and then you can do the cross correlation to see, hey, am I better off shopping with my partner? work together, or do we just sit there and fight? No, I don't want to have that for dinner. We had that for dinner last night. And so the cross correlation is sensitive to the motion or the position, in this case, the image correlation of both dyes at the same time. So it's the same sort of formulation, but now instead of taking the same function and taking a copy of it and shifting it, you take one image, you take a second image, and you see how well they overlap, how well they correlate. And that has the advantage that now the amplitude has a positive correlation with the number of molecules that have both fluorophores. And the nice thing about RICS, of course, is that you can see it in real time, in motion dynamics, so you can not only see that they're interacting, but you can see how fast they're interacting. Is there a change when they're interacting? So here's GFP, here's M cherry. We're separating them, or we're expressing them individually in the cell. And here we get our nice autocorrelation function. We can fit it well. We can determine how fast the GFP is, and the M cherry as well. And now we do the cross correlation, and there should have been none. But we have some. And this is the thing about correlation spectroscopy. Correlation spectroscopy is absolutely great. If it correlates, you'll see it. It's also very dangerous, because if it correlates, you'll see it. So you have to know what you're looking for. So in this particular case, I mean, this is not GFP and M cherry, but it's some dyes. You have this little thing of spectral crosstalk. The fact that there is a small amount of green photons that will be detected in the red channel. And it's just, you know, until Lucas can finally make us very, very small fluorophores without this tail, we have this problem. And even if it might be 5% or 3%, its correlation says, hey, I see something here, and when I see something here, I often see something here in the green channel, and it correlates. So you can correct it. You figure out what the crosstalk is, how bright your signal is. That's complicated. We'd prefer to do better measurements or try to find more elegant ways of getting rid of this. And so that's where pulse interleaved excitation comes in. And so the idea is basically, instead of just doing one laser or both the green and red laser on at the same time, you would now alternate between them. This actually came from the group of Schiem and Weiss. They were doing something called a microsecond ALEX, and then, you know, I heard the poster presentation in 2002, and I thought, wow, I like that poster. It's really interesting. I have no clue what it's good for, but it's kind of nice. And then later I actually thought, well, actually for correlation and getting rid of crosstalk, this would be a nice idea. So what do? We have a clock. We fire then the green laser, perhaps on the clock. We fire then the red laser shifted. So we're doing two experiments at the same time. We're doing a green measurement, and then shifted by about 20 nanoseconds, we do the red measurement. Right, so you can see then, and then we have two detection channels, the green detection, we have the red detection. So you see, there's a photon in the red channel, and that came after the red laser. Here's one in the green channel after the green laser, and then here's one in the red channel after the green laser. And so if we now look very carefully at when all of these photons arrive, so we start building up statistics. Here comes photons. When did they come with respect to the laser pulse? And I start making histograms. And at the end, I end up with some sort of nice TC-SPC, time-correlated single photon counting histogram, showing me here's the intensity of the green after green excitation, of the red after green excitation, and here the red after red excitation. So we have pulse lasers coupled into fiber, 3 different detectors. And now we get what we call the pi channels. So we can now separate photons based both on which detector it sees it and which excitation source it belongs to. So we separate them out. The crosstalk photons end up in the red channel with green excitation. So we just correlate green with green excitation with red excitation. Correlation disappears. Beautiful. I mean, we were doing some single pair FRET measurements, and my students were having really hard time finding double labeled molecules. So I told him or them to just do an FCCS measurement, and less than 0.1% were interacting. So it's like, okay, it's not labeled very well. Good, so if we now do pi, this cross correlation disappears. If we make a tandem construct where they should be co-diffusing, the correlation stays, and they do correlate. Good, so what can you do? Well, we can do crosstalk-free imaging. So we beautifully get rid of the spectral crosstalk. We can do FRET imaging. So we can look at the FRET signal and then look at what the acceptor signal looks like independently. We can actually, if you want to do correlation spectroscopy and you have FRET, that becomes also complicated, but we could actually add the photons back. With the FRET photons, we can add back to the green channel and get rid of the FRET effect. So we can do some really nice things. And what's also interesting, okay, we can do here, as I mentioned, for getting rid of spectral crosstalk, we can take the green channel to green excitation, red with red excitation, get rid of the cross correlation in RICS, but we also purify the species of the autocorrelation. Because what happens if you have here crosstalk, the crosstalk is a different species. So you have a bright species, your correct green species in the green channel, you have you have the red signal in the red channel, but you also have this crosstalk of the green that's weak in the red channel, and that sort of messes up. It's a two species problem. You get this this squared fractional intensity dependence, and that messes up the amplitude, which also messes up your determination of how many molecules you get. And so you can correct that by using this pi. And like I said, you could also now correct for FRET by adding them back together. You still have direct excitation to deal with, but that's the way it is. Now we have pulse interleaved excitation. We're using sub-nanosecond, actually about 100 picosecond pulse lasers. And there was this idea by Jörg Enderlein of using the lifetime information for doing correlation. So you can imagine now you have two dyes, Alexa 647 and Ado 647N. They have the same spectrum, but different lifetimes. Can we separate them? Well, in essence, we can do a probability distribution. The one with the fast lifetime component, well, that has a high probability of being species 1, less of species 2, and then we get to lower times, we have a high probability of the photon coming from species 2 and not species 1. So we can weight those. So you let the math do the job. I think I have to skip the details. Probably makes most of you happy. But we can actually then, by multiplying weights, separate out from the same signal species 1 versus species 2. So with this was originally done to get rid of some very ugly after pulsing. So you had one distribution that was flat of when the photon arrives from after pulsing, and then you could get real signals out. You can also get rid of scattering. Scattering is basically the IRF of your system. And this happens when you go to very low concentrations. So if you dilute your sample, when you start getting down to low picomolar concentration, even with the best filters, you still get photons, scattered photons from the laser that start to compete with the amount of signal you're getting from your fluorophores. And so actually then that turns to the fact that you start really miscalculating how many molecules you have. If we now apply the filter, we can get rid of the scattering and we get the right amplitude for the correlation. And here's actually the measurement of Alexa 647 and Ado 647N on DNA. If we measure them alone, we see what they look like. If we measure them at the same time, we use this fluorescence lifetime imaging. We can actually separate out the two components. We can also do the cross correlation. They don't cross correlate. And so this works very well. And so we can do this, what we call Raster Lifetime Imaging Correlation Spectroscopy, or RELICS. And here we actually wanted to separate out fretting species from non-fretting species. So you have a short fluorescence lifetime of the donor when the acceptor is present, and when the acceptor is not present, then you have the donor-only signal. We could get spectral weights and thereby significantly decrease the amount of cross-correlation without FRET. There should be none. This depends very sensitively on your filters. Okay, so what happens if you don't have lifetime information, but you have spectral detection? Can we do the same sort of thing? Can we use statistics and mathematics to actually filter the species? And so this is, again, from Jelle Hendricks. And so now you have a number of images, so maybe 34 different images. You have so you have intensity in each of these channels. And we can make spectral filters to separate out, for example, GFP and M-Cherry, and we get rid of this cross-correlation due to spectral crosstalk. Can we push it? Well, yes, we can actually do this with GFP and Ado 488. Their spectrum is very similar. we get the nice weights and we get rid of it. The cross correlation goes to 0. If your weights are a little bit wrong, then it's not perfect. That's one of the challenges of this. Then we also played a little bit of how much can we shift the different populations. If they're both equal populations, you do fairly good. If one is 10 times more than the other, then of course at some point you can't correct the one species very well. And then we actually said, well, do you really need all of these channels? We actually started separating now for 3 species, 3 different channels. So 1 channel per species, and as long as the spectral contribution in each channel is significantly different from those 3 species, we could separate it. So here we had Venus, GFP with M Cherry together, 3 species. We can look at the cross correlation between GFP and M Cherry, it's there, and between GFP and Venus and M Cherry, it's not there. So with this spectral imaging, you can also get rid of it. It's been this algorithm has been incorporated into the Zeiss software. And with that, I get this signal that I'm supposed to stop. So all the really cool science that I was going to talk about, where we applied this to viruses, I skip over and just come to acknowledge my group for the excellent science. And a lot of good people I've had to work with, the collaboration partners that I've been talking about in this talk, and the funding, and thank you for your attention.
Thanks, it was a very nice introduction to RICS. I have 2 questions. One about the technique. How robust is against optical aberrations? For example, would it work in deep, thick samples?
Okay. I guess I have that, I can't answer that really directly in the sense that I can't convince my students to misalign the microscope. They want to take good data, right? But in essence, you have the correlation curve. The correlation curve is fairly, let's say it has a fairly non-sensitive structure. So I think you can be misaligned a bit without having a big impact.
But if your BESFs are like potatoes, that would start...
I mean, if you have something that in 2 dimension looks like a potato instead of in the 3rd, that it would make very ugly looking shapes, yes. And then you would mess it up.
And the second question is about the biological application. If the the condensates, sorry, I forgot their name. The chromocenters are condensates. Do you have an explanation of why they are not spherical and they have this very non-spherical shape?
So they act liquid-like in the beginning, and then they shift to more solid gel-like in the 4-cell stage. And so that's why we lose the circular structure later.
Okay. Very interesting, thanks. So you showed this approach where you can have an arbitrary mask and look at the diffusion within this mask, and you can look at different regions. I was wondering if you can do the reverse, where you would take, let's say, the whole nucleus, and you figure out you have a sum of different behaviors, let's say, binding and diffusion. And can you, for instance, start with a random mask and optimize the mask to make some of these behaviors stand out, to figure out where it happens in the nucleus? For instance, you would try to optimize your mask to have as much binding as possible within this mask as to figure out where it binds.
Good. I don't know. In theory it should work. It sounds good. I mean, it's an optimization problem, I imagine. I mean, the point is that when you first select, you're averaging over your image that you select out. And so you really have to see whether you have something. But if you have something you can start with and you start to optimize, it's an optimization problem and should, in principle, work.
Yeah. Good. Thanks.
Hey Don, thanks for the great overview. I have a question about the RICS implementation. When you're, as you know, I'm recently into things like resonance scanners, where your scanning is non-uniform.
Forget
it. Right. Anything? No, just...
Yeah, I mean, it was very interesting when Enrico and Michelle came out with RICS. you really learned which manufacturers made good scanners and which really did a lot of crap in the background so it looked nice afterwards. Because the way it works is you assume that each pixel has the same distance and the same time. And that's the same regardless of where you are in the image. And if that's not true, then you start messing up with your correlation, because it's no longer, you sum over all of the image, and if the left corner and the right corner are not the same, it messes up the function.
And so then, if I'm faced with a random confocal, how do I know what is a good one? And tonight over a beer, can you tell me which companies make good ones for RICS and which ones don't?
Actually, I think a lot of the manufacturers have come a long ways in the last 15 years. So most scanners right now are good, but the resonance scanner, of course, your whole idea is you push it in resonance, and so you have a sine curve in the end. Now, what you can do there is you can take the center region. The center region is still fairly linear, and if you cut it to that region, it will still work. You just have a smaller gap in the lines.
Yeah. Thanks.
Hi, just a very naive questions. So you showed for the condensate, the different diffusion outside and inside. Would you think that FCF can be used to specifically look at the intersection, the boundary, and look what could be, the possibility for what you have inside to go outside more specifically with the resolution that we have?
In theory, yes. In practice, I think it depends on the system. So in our system here, our collaboration partners brought over the mouse embryos at the two cell stage. Some were working well, some were working less well. We ended up having to measure a lot of cells to get good enough statistics to see the differences we were looking at. And in that case, if I wanted, there is this idea, I guess, to explain a bit the question. You could do what's called pair correlation, where you look inside, outside, or scan. You could actually see the time scale it takes to go from inside and outside. You can do that, but then I would, if you go to an in vitro system, that should work well. I think in cells it's much more complicated, just due to the fact that how stable the sample and the biological heterogeneity it is that so you have to measure an awful lot to see small differences. Okay.
Yeah. Sorry. Just a question.
You want me to go through the virus stuff? No problem. We have another 5 minutes. Yeah.
I know the story indeed. So no, I was more thinking about. Can you comment about one maybe of the drawback of RICS when you have to face two diffusion coefficients that are very far,
right,
one from the other one? So
right. So RICS is fairly invisible to triplet stuff because that's just too fast, and so that gets averaged over, which may be an advantage. You have two dimensions, so it has the advantage that you have a fast axis and a slow axis, so you cover a range, and you can change the scanning speed to correlate to what you're looking for. But it is a fairly fixed size, so we can distinguish between what we call a diffusing species and immobile, which is just the point spread function. That works well, so you can get two. But that means that what's moving slower, and I would have shown that had I had time. But anybody's interested after beer, we can talk. So we could say, okay, we could fit what's immobile. Again, immobile means that it's not moving on 50 millisecond time scale, so it still can move. But if you're looking for separating out diffusion coefficients that are different by a factor of 5 or so, then I think I wouldn't use. Well, even FCS. The decay is not all that sensitive, right? And so. The nice thing is you can get an average diffusion coefficient. Of course, if you know one well, you can fix the one and then you can fit it to a model. But that's the whole problem, that the decay of the function is fairly slow, and so it's hard to separate things. You need big differences in diffusion coefficient to separate them out. You can put in an anomalous model and it will flatten it out a bit. You can do that.
Two last question.
Thanks for your talk. Do you have any feedback about some conditions where the main assumptions will maybe not hold, like the stationarity of the process and this kind of thing? for the fact that it's uniform also, and if they do not hold these assumptions, the main assumption behind this thing, do you know how to detect that they are not respected? Like, for example, if you have a diffusion which is changing over time in the same area,
I mean, if it's not stationary, right, then you have to somehow measure fast enough. Now you have the advantage.
but how do you detect that? That was my question then.
Oh, how do you detect that?
How do you know that it's not holding this assumption?
Oh, experience. Experience is what you get when you don't get what you want. You know, I guess it's, yeah. I mean, that's difficult. I mean, in essence, that's what you tried to do. Is I mean, it's similar to, I guess, the kappa squared and Fred. You just sort of average over. You say we just average over all possibilities, and you get the average value out in the end. I mean, typically, what you need to do is when you're doing the scan, you want to have, you know, the molecules moving around somewhat during the scan. But when you come back for the next scan, it should have totally been moved away. And of course, there's a lot of tricks which I didn't talk about. If you're measuring in cells, then you typically do a moving average window of like 3 frames to get rid of cell movement and other things, so you can look at, you know, really the faster motions. And you know, so there's typical, depending on whether you're looking at diffusion of proteins in the membrane or in the cytosol, you can use different scan speeds, and that's the paper that Jelle had with Longfils in the Biophysical Journal. They went through the optimal parameters, and that would be a good place to look to start with. So that's kind of nice. You can adjust the scan speed for what you want to look for. If you want to look for things that are changing in time, well, that's more difficult. Then I think you'd have to do ICS with talk to Thorsten, and then, you know, the AI will pull it out in 5 frames, right? Or 2500 anyway.
Thank you for a nice talk. Just a short question, stoichiometric wise, you see pair interactions blue and yellow, but how about 2 blue with 1 yellow, spatiotemporally correlating? If you take into account absolute intensities, can you also say something about like multivalency for a clean sample? I think it can work, but how well can it work for
Of course,
mixed?
we can do everything. No, I mean, the one example I showed here was 2 quasi-blue, right? Venus and EGFP with M cherry, right? Okay. So in essence, we see the GFP does correlate with the M cherry. Venus and GFP do not, and Venus and M cherry do not. That's what's shown down there.
But for this, you need like 90% of.
I mean, okay.
Like how well does this ratio have to exist in order to be able to still detect it?
Okay, that's a question of sensitivity. I guess if you look, the correlation we're getting here is almost times 1, you know, 0 or 1 times 10 to the minus 2, and the noise level here is maybe 1 to 2 times 10 to the 3, and so we have a factor of roughly 10 or 5 that we can deal with. And the fact is, of course, as you know, M cherry maturates about 40% of the time, and you know, GFP 80%, as you mentioned. So under those conditions, we can see things. We do have a little bit of room. I mean, it's with pi. It's really amazing how well you can get 0 cross correlation. You can get rid of the crosstalk, and you're very sensitive to the interactions. It's a question of just then how long do you measure, and you can always, you know, if you have a nice system that's behaving, then you don't have to do it in 100 frames. You can do it over 300, 500 frames and get better statistics.